Liebe Kolleginnen und Kollegen, es freut mich, Sie begrüßen zu dürfen zum heutigen Oncolunch. I will have to switch to English now because we have a very special event. First, I would like to have you sign here and have your confirmation. We will thus have the presentation by Professor Ajani, Jaffer Ajani, his correct pronunciation, from MD Anderson. But before he will speak, Ms. DeGregorio from the DeGregorio Family Foundation will give a short introduction to this foundation, which is from a family afflicted with gastric cancer. So please come up here and
let me know to that later. Thank you very much for the introduction. It's a pleasure to be here today. I want to introduce you to the DeGregorio Family Foundation. It was founded in 2006 by me. I'm president and active fundraiser. During the day, I work on Wall Street, and my journey from going from Wall Street to serving as president of a foundation began very painfully with a history of gastric cancer in my family. And it turned out we are one of the 200 or 250 known families with a mutation on the CDH1 gene. My father, who is a radiologist and actually got his degree at University of Lausanne, came from a very large family. And I spent my life watching that large family become a very small family. Every 18 months, we had a relative who was diagnosed with stomach cancer, and the journey from diagnosis to death varied, but it was always tragic and painful. From the time the person died, 18 months would go by, and the next person would be diagnosed. We knew it was hereditary, or we speculated it was a family curse. At that time, nobody was sure, which. Each time a person died, I was fearful that my father would be next. And unfortunately, in 1976, when he was 50, he was diagnosed with gastric cancer and passed away 16 days after diagnosis. In 1980, my father lost a cousin as well, and that was the last person that passed away. So we sort of thought the next generation was immune to this disease. We asked, my brother's a physician, my sister was a PhD in psychology, I was a non-medical person, but we all had stomach issues. My sister had bloating, my brother had acid reflux, I had heartburn. So we would ask the doctors constantly, with this terrible family history, should we be worried? My brother asked, should we all be screened? We were told, no screening necessary. I was told, people in the US, they don't get stomach cancer anymore. So we didn't worry about it. In 2003, my sister started with the so familiar symptoms. She said, gee, I'm losing weight, I'm not even trying. I'm full on only half a sandwich. And I was like, it can't be. So we begged her, go to the doctor. She went 9 months with symptoms as bad that would send her to the emergency room. She was sent home with a diagnosis of gas. Finally, she was diagnosed with ovarian cancer. Apparently she had a Kruger tumor, and ultimately they found out it wasn't ovarian cancer, but gastric cancer. She passed away 10 months after diagnosis, and when she passed away, we found out that she had a CDH1 mutation. So my brother and I were like, how could 27 years have gone on and nothing nothing personal to the researchers. Nothing had really changed as far as, not being able to be diagnosed, being diagnosed, in stage 4 when it was too late. So we mustered up the guts. We got tested. My brother was negative. I was negative. And at that point, we sort of decided we kind of have a moral responsibility here. When I looked into what type of funding there was available for research from private foundations, foundations would start, and then a couple of years later, they would sort of go away. I looked into what the NIH was funding, and it was $8 million for esophageal cancer and like $12 million for stomach cancer. Those numbers have gone up, but not substantially. At that point, I decided we're going to start our own foundation. And, with our foundation, the goal of our foundation is to be the catalyst for researchers to get other funds. I know we can raise money, but we're never going to raise the amount of money that's needed for all the research that's going to be required. So what we've done is, once a year at this point, we fund a grant. We announce it in August. Grants are due October 31st for a January 1 funding. This year we're offering $250,000 to be funded over 2 years. And we hope, as we continue to be successful fundraising, that at some point we'll either do grants more often or we'll do grants for larger amounts of money. So far, our successes have been, in 2006, when I began the foundation, through friends and family, we funded the gastric cancer registry at Memorial Sloan Kettering. We funded that for 3 years. In 2008, I went out to the public for funding. And at that point, we funded the gastric cancer registry to be abroad. They're open in Nigeria, and they have several other sites that they're looking towards. We also issued a grant to Dana-Farber of $100,000. They actually were able to secure $1 million from the NIH after our funding. They just published in Cancer Research in August from that research. In 2009, we actually didn't fund a grant because we didn't have very good turnout for how many grants were sent in or the quality of the grants. That was actually 2009 for 2010. 2011, we gave out $250,000. It ultimately was split between Dana-Farber again and Cornell. And we're issuing our grant now. We're in the grant process for October 31st. As I look towards the future, I'm here because I want everybody to know that we are available for funding. And we're trying to increase the amount of grants that we receive. So thank you very much for your time. And I have some brochures if anybody's interested.
Thank you very much, Ms. DeGregorio. Is there any comment or question at this time? If not, I'm honored to introduce Jaffer Ajani from the MD Anderson Cancer Center. He has a sort of American dream career, starting in India and doing his medical training in India, and then coming to Pennsylvania, Altoona, Tulane University, and then as a clinical fellow to medical oncology 1980 to the MD Anderson, where you have been since then. And of course, you're known to most oncologists here, and I think you have been the chief of the Department of GI Oncology at some time at the MD Anderson.
Sometime, yes.
At some time, yes. Now we're looking very much forward to your presentation about the biology of upper GI cancers. Thank you very much for coming here, and thank you for supporting this event.
good afternoon, and I'm very honored to be here today. And after the presentation, I really appreciate what you're doing for the research community because we do need help. So I like to first acknowledge many colleagues from basic science and clinical sciences that have helped to move the field forward to some extent. And we are also receiving funds from the NCI. We have 7 NCI grants, some MD Anderson grants. MD Anderson has actually given us close to $1 million to do research in gastric and esophageal cancer. And of course, many generous donors. So what I was going to talk about is predominantly molecular biology from a clinical standpoint, because I think I'm just going to talk about my own perspective on what cancer is all about and how treatment affects cancer, and then what we have to do. So I think some of the subjects that I'm going to talk about, some of you know more about it than I do, but I'm just going to tell you my perspective. And I think we need to have a dialogue in order to really decide what is the best future research. So just at a basic level, we are all composed of this code in the DNA, and that's what drives the cancer as well. So the classic paradigm is that DNA has the code that is then, I'm sorry, but maybe the pointer is not going to work. So DNA has the code, which is then transcribed into RNA, and then the protein is made at the bottom. But I think there is some new information that suggests that the process is not all that simple. And that the way we have been interrogating cancer is probably not ideal. We need to understand a little bit more. So,
use the mouse if you want.
Yeah, would you hook up? So, you're probably familiar with this particular project called ENCODE, which is the Encyclopedia of DNA Elements. And just about 30 papers were recently published. And I think it is very revealing because it has so much new information that I think we will have to change the way that we do some of our research. So what is found in this project is that there is the 76% of our DNA is functional in a way that we never understood it before. So what we understood was 2 to 3% of DNA was coding for protein, and previously rest of the DNA was thought to be junk. Then it was thought to be microRNA genes, and now we know through this effort that there are many portions of DNA that are actually regulating the entire coding DNA. So what that means is, if you're just looking at the DNA changes in the coding regions, that probably is not sufficient because you have mRNA, and then rest of the DNA is regulating. So there is RNA regulation after transcription. So, and we have been ignoring that. So the proteins are formed only after RNA regulation. So that step is becoming more important. I have another picture to show you later on, but what this ENCODE project, which was funded by the US, $228 million, or I'm sorry, $88 million. And what is found is that there are more coding genes than we thought before, about 500 more coding genes. But what is surprising is there are 18,000 RNA genes that were previously not well characterized, that are present in our DNA. There are several other aspects of the ENCODE project that seem to be very interesting. One is the physical configuration of the chromosome, and there are regions, as shown on this side here, that there are interactions between the DNA which is far away from each other. And the reason it can interact is because of the physical configuration of the DNA, the way it folds, and three-dimensional interactions. So you can have a gene over here, but the enhancer region can be way further down. But in reality, it is folded in such a way that they are close together. But there are many, many other interesting things, the way transcription factors work. And then there are these DNAs, hypersensitive sites that have been discovered that are really specific to the type of tissue. So liver cell will have a different DNA configuration as well as these hypersensitive sites as opposed to your retina, for example. So I think we have to learn all these for cancer because the cancer is, again, the transcripts are being regulated by RNA, and then you get the proteins. But there are many, many things about cancer that are different than normal cell. This is just the normal cell, and on your left, when there is a lot of nutrition, a lot of glucose, then the apoptotic machinery is shut down. There is no need to commit suicide. But on the right, when there is glucose deprivation, then the cell either will try to survive with autophagy and other mechanisms, but ultimately it will commit suicide. But that's the normal cell. But the cancer cell doesn't seem to do that. So this is again the normal cell. And here you have a glucose molecule on the top, and it will produce some protein and some nucleic acids, but normal cell is really not under pressure to make anything other than ATP, just the energy to carry out its usual function. On the other hand, the cancer cell is quite different because of the growth factor on the right side. The growth factor then leads to upregulation of many oncogenes, and those oncogenes will modify the metabolism. But we have to remember that cancer cell can only think of one thing, is to multiply, in addition to migrate and so on. But it needs to create the biomass. It is not really interested in having a lot of ATP around. It is interested in having a lot of nucleic acid around, a lot of amino acids, and other components such as the lipids in order to make another cell. So it wants to divide into two, and so it has a very different metabolism than the normal cell. And it must use some amino acids in order to have the source of nitrogen. Because glucose does not have nitrogen. So we will come back to this at a later time, just in a few moments. This is a busy slide, but essentially, if you have just glucose as an energy supply, the cancer cell cannot survive because there is no nitrogen, source of nitrogen. So it must use glutamine or glycine or even some other amino acids that must be used in the synthesis of different components in order to have enough biomass to multiply into two. This is why the cancer cells, we are told, are fatter, and they have more iron, and they have more products than the normal cells. So on your left is the normal tissue that is differentiating, and you can see that the pyruvate utilization is higher than the lactate. Lactate is actually excreted out of the normal cell. On the other hand, the tumor can function in the presence or absence of oxygen. But there is a higher accumulation of lactate, and then lactate is used in the Krebs cycle to produce other components. But I wanted to point out the efficiency of normal cell on the left. If you look at the bottom, you have 36 ATP molecules that are being made from one glucose molecule by the normal cell. But the cancer cell makes only 4. And the reason for that is, as I mentioned, it's busy accumulating the biomass rather than energy. So the next thing I want to mention is the oncogenes that are utilized by cancer, and then go on to demonstrate that cancer cells can utilize multiple oncogenes. It's just not, we talk about oncogene addiction, but the oncogene can, this cancer cell can utilize multiple. And I want to show you 2, 3, 4 examples. And this should alert us that if we treat one gene at a time in a patient, we're not really making big progress, because the cancer genome is much too smart for us to manipulate one gene, and then we manipulate the second gene, and we're just chasing this very sophisticated machinery. So the blue bar, very high bar, is the KRAS oncogene that is mutated in a lot of cancer. You can see the blue in many, many cancers. And the P53, the tumor suppressor gene, is either mutated or suppressed in a lot of. So those are the 2 genes that are commonly abnormal in many solid tumors. So the question is, why cancer cell wants to use KRAS? Well, what can KRAS do for cancer cell that it has? It is mutated in so many cancers. So just to show you some of the things that KRAS can do, this is located on chromosome 12. And the red line, you can see that's the KRAS. And this is the primary structure of the protein. It's a very small protein, about 189 amino acids, with 3-4 domains. And this is the tertiary structure with the helices and beta strips. So this is a globular protein. So one of the things KRAS can do for cancer cell is allow the cancer cell to progress through multiplication from G0 to S. So normally, as you know, if there is DNA damage in a normal cell, it will shut down all the checkpoints, and it cannot progress to multiplication. And that's because a P53 wants to repair all the damaged DNA before multiplication takes place, because we want to have healthy cells. But for some reason, which I don't understand, there is tolerance of DNA damage in cancer cell. Cancer cell can tolerate the damaged DNA and still will continue to multiply. And one of the reasons is the KRAS. So KRAS will allow the cell to enter G0. It will block TGF-beta, which is a tumor suppressor gene. It will block P27 and retinoblastoma gene. So all these genes that are trying to prevent the cell to multiply because DNA is damaged, KRAS will facilitate that. And ultimately, a cell with a damaged DNA, with a lot of mutation, can make a copy of itself. So this is one reason cancer cells are using KRAS. Another reason I sort of mentioned is shown on this slide. So you have the necessity to make amino acid, nucleotides, and proteins and lipid when you have energy source, such as glucose and glutamine and glycine. So KRAS modifies the metabolism of cancer cell to allow it to produce the biomass. So this is the second reason why KRAS is preferred by the cancer cells. And this is a busy slide, but this is another reason KRAS can help the cancer cell is producing pro-angiogenic growth factors. It actually facilitates angiogenesis, and then it will allow recruitment of normal immune cells such as macrophages, which basically help the tumor bed to secrete other growth factors, including angiogenic factors. So if you think about it, then KRAS is central to cancer cells, such as in solid tumors, where it will allow nutrition, it will modify metabolism. So I'm going backwards. So you have all the nutrition you need, you change the metabolism, and then you continue with multiplication. So KRAS can do all these three things. But there are other oncogenes that are also able to do this kind of work. So now I want to go on to 2-3 other oncogenes. One is HER2/neu, and I think many people are more familiar with this than I am. So this is the primary structure of the HER2 protein. It's a very large protein compared to KRAS because it has 1,255 amino acid, many domains, because it can do all kinds of things. This protein in physiologic states works in variety of tissues. This is the tertiary structure. And the drug trastuzumab is important because it can prevent the function of HER2. So HER2 doesn't have a ligand, but it likes to hook up with HER3. And the reason it likes to hook up with HER3 is because of PI3 kinase. So there is a P80 domain that will phosphorylated by HER3, and then it has the survival signal. So HER2, although it can dimerize with another HER2 protein or HER1, the preference is really with HER3. And what trastuzumab can do is hook up to the extracellular domain on the left of the right picture and will prevent HER3 coming in contact with HER2. And so this is just a simple mechanism of action of HER2, but it is more complex. It's much more complex than that. So this is in breast cancer model, where you have the tumor cell with HER2 protein shown in black, a little strand, and then you have the antibody trastuzumab hooked up to that. So this is people use this kind of slide to demonstrate that there is HER2 or oncogene addiction. So if the cell is dependent on one growth signal, and if you block it, then the cell will, what we can call it, cell will panic. So there is a panic mode through which it will release a little polypeptide, which is the HMGB is considered a polypeptide by the immune system. It's a danger signal. So immune system will pick that panic signal as a danger signal, and the antigen presenting cell will recognize that particular string of amino acids, and it will take it to the T cell, and I will explain that a little bit later on. And the T cell will come and kill that cancer cell. And of course, the antigen antibody complex is cleared by another immune mechanism. So in reality, the way trastuzumab works is to stimulate the immune system, which kills this cell. But we know that some of the cancer cells will survive trastuzumab, and they use variety of mechanisms to survive trastuzumab. And this is the problem of cancer we have. So the cells that can survive trastuzumab can do all kinds of tricks. And this is a paper on breast cancer model. It's published from MD Anderson. And what you see on the left side is you have the 4 and more proteins on the these cell surface proteins, trastuzumab hooked up to HER2, and there is resistance to trastuzumab because the cell is now using other oncogenes. It's using EGFR1, IGF1, it is using c-MET amplification, or it can do all sorts of different things. So it is basically reaching out to all other oncogene machinery it has, because all it wants to do is multiply and do other things. So what was found is that in the process of acquiring resistance to trastuzumab, there is hyperactivation of the SRC protein, which is the intracellular protein complex. And through this hyperactivation of SRC, it can then engage other oncogenes. So if you see on the right side, if you block the SRC protein, you can make the cell again sensitive to trastuzumab. So I actually treated one patient who was sensitive, whose tumor was sensitive to trastuzumab, but then became resistant. I tried lapatinib, and the patient's tumor became resistant. Then I continued on trastuzumab and gave patient dasatinib, which is a SRC inhibitor, and that patient had a dramatic response. I tried that in another patient, it didn't work. But this is just one example of how cancer cell can become resistant to manipulation of only one gene. So we're basically manipulating one gene at a time. But if you manipulate 2 genes at a time, which is on the right side, then you get some advantage. So the other mechanisms by which the cell can sense that there is a problem, that means trastuzumab is around and it is bothering the cancer cell, it will activate one of the proteases genes, and it will just cut off the extracellular domain. So just clipping of the receptor, rest of the receptor is constitutively active. So then the signals are coming in. There is no need to have extracellular protein. So this is a known mechanism. It's not a mutation. The actual mechanism by which this cleaving takes place is not well known, but it's not a mutation in the HER2 gene. It must be what I was talking about, the RNA regulation. So there's somewhere in the DNA that knows what is going on in one location in the cell, and it will find a way to bypass that. So this is an example in breast cancer where the breast cancer can simultaneously, a breast cancer cell can simultaneously have a truncated protein where the extracellular domain is gone and a full receptor at the same time. So this is the argument used that it's not a mutation in the HER2 gene. So it may be possible to take advantage of the structure of this protein. So there is a molecule that can affect the truncated protein, but you can see that there are many places where we can interrupt this machinery. Again, dealing with only one gene at a time. So this is like a composite of the HER2 resistance. And you can see different mechanisms by which we can try to interrupt. So TDM1 is something that I want to come back to, but we can interrupt extracellularly, intracellularly, or through emergence of resistance, we can manipulate the resistant genes. So another example I want to give you, this is our own work in gastroesophageal cancer cells. An example of how manipulation of one gene is probably not sufficient. So this is the hedgehog pathway, where you have the surface, two surface protein, the SMO and the patch, and ultimately the GLI1 comes into the nucleus. So this is sort of a traditional embryonic pathway through which cancer cells tend to multiply. And the reason they use embryonic pathway is because embryonic pathways are highly efficient for rapid multiplication. So cancer cells often engage those genes because they are already trained machineries. So it's very easy to just grab one of those genes. So when we were doing some experiments, we exposed the cells to TNF. So that is a cytokine, an inflammatory molecule, and it should engage the mTOR pathway. But what we saw was in 3 different types of cells that the GLI1 was upregulated. So if you stimulate the cell with TNF, we don't expect GLI1 to be upregulated. And on the right side, you can see the mRNA in GLI1 components are different sonic components are upregulated. So TNF is stimulating sonic pathway, which was kind of a surprise. And initially we didn't understand. So if you interrupt the mTOR pathway with rapamycin, which is the RAPA++, your GLI1 is still very active. There is transcription factor in the nucleus. And so this suggests that if you inhibit mTOR pathway, that's not going to be sufficient. So we took the patient tissue and looked for both markers, both pathways, hedgehog pathway, which was the GLI1, and also looked at the mTOR, which is the phosphorylated S6 kinase. And they are both upregulated. So what that suggested to us that maybe if you inhibit the hedgehog pathway, the cell somehow likes to use GLI1 because it's embryonic in nature, and it will get the GLI1 from somewhere. It's not the hedgehog pathway because you already blocked it. So we use the inhibition of the hedgehog pathway and mTOR pathway, which is G and R, and you can see you get much more advantage if you combine the two. And these data, there's lots of data published in this paper that I'm not going to discuss, but this is just an example of double inhibition. So just to summarize, on the left side is the canonical embryonic hedgehog signaling, where you have 2 proteins and then the dissociation of GLI1 from SUFU, and the GLI comes into the nucleus, and then it stimulates a lot of genes, and that is survival pathway. But if you block with GDC molecule, that's the one we used, if you block it, then you shouldn't have GLI1 in the nucleus. And yet we found GLI1, and then what we found was that S6 kinase, which is stimulated by TNF, actually hooks up physically with GLI1 and phosphorylates GLI1 to break that complex, and you have GLI1. And that is why we need to now inhibit both pathways, HOX and mTOR, at the same time. And this is the trial that I'm hoping to do through the National Cancer Institute, through a grant. Finally, I just want to show you one more example. It's a beautiful example of the BRAF mutation, the same location in two different tumor types, but the drug works only in one tumor type. It doesn't work in both. So there is a BRAF inhibitor, and the first panel on the lower side, that's the melanoma cells, and you can see the control is black. If you inhibit EGFR pathway, it's still black because there is no effect on the cells. But if you block with the BRAF antagonist, then the plate is clear. On the other hand, on the right side of that panel is the EGFR transfected in melanoma cells. So you have BRAF mutation and you upregulate EGFR. And there, with the double inhibition at the bottom, you get the advantage. You don't get the advantage with BRAF inhibition alone. And the reason for that is when you have EGFR and BRAF, both are active. And if you block BRAF, the cell will use EGFR as an oncogene. So that's a bypass mechanism. So on the right side is the colon cancer, where we know EGFR pathway is often active, and BRAF mutations do occur, the same location as melanoma. And here, only the combination seems to work. And this is concentration dependent, but the BRAF inhibitor alone does not seem to do anything. So this is now borne out in the clinic, where people took patients, colon patients with BRAF mutations, and gave them BRAF inhibitor, and there was hardly any response. But now people are busy combining the two molecules, and I think there will be some advantage. And this is, I can skip this slide because this is just the in vivo xenograft data. So in melanoma on the left side, if you inhibit BRAF, you get tumor regression. And in colon, you don't, and that's because of hyperactivation of EGFR. So I think we can say that cancer has this very sophisticated genome that evolved over 3.5 billion years, survived through all the evolution and selection, and we are dealing with that genome. So if we keep manipulating this genome, it's just going to sidestep. So you inhibit one gene, then it uses another gene. You inhibit that gene, it uses another gene. And then the cancer cell becomes resistant and the patients die from cancer. So should we just continue to do this kind of approach? You constantly find more targets and then manipulate the cancer cell, and it will always outsmart us because we can only give so many drugs to the patient because of toxicities, and all the pharma is fighting with each other, all the contracts and intellectual property. So we can see that we are not going to have tremendous advantage if we just take one biologic, two biologic, three biologics, and we're just chasing the cancer. So why not consider something like this machinery here, where we could kill the cancer cell? If you can kill the cancer cell, then you don't really have to deal with all the manipulations, all the DNA smartness that cancer has. So just the next 5 minutes I want to spend on this. So here is the APC on the right, and that's the T lymphocyte, and they are communicating with each other. So APC has a message for the T cell. And so there are many ways of killing the tumor cell, not manipulating the tumor cell, but just killing it. And they are shown here, and I'm going to kind of repeat some of that. But there is a much smarter way, which is on the upper left panel. If you look at the bottom, the conjugated toxin is probably the best way. There are many ways to manipulate the immune cell and the cancer cell, but that appears to be the best way. And then on the right side is where we can train the immune system to kill the cancer cell. And I think there's a lot of hope on this side. And then the final example is where since the tumor is recruiting host cells, we can deplete the host cells so the tumor doesn't take advantage of the skills of the host cell to promote itself. So this is the T cell, and as all of you know, that T cell is the mastermind behind immune action. So the APC that are resident in the tissues or they are circulating, once they find something that they think is abnormal, they will bring it to the T cell. They look all over the body for the right T cell and give it to the T cell, and it's up to the T cell to decide whether to take the next action or not. The APC commits suicide after it talks to the T cell. It undergoes apoptosis. So as far as we know today, there are these receptors. There are 6 receptors on the right side, and there are 6 receptors on the left side, and these are the inhibitory receptors. These have evolved in order to suppress the autoimmunity. So all these receptors are very busy in our system, and otherwise our immune system will constantly attack our normal tissue. And so in cancer situation is the same. The cancer cell can have abnormal protein on the surface, but the T cell, through these receptors, will tolerate the cancer cell because there are some similar features to the normal cells. And this is why the cancer cell can happily divide in a patient with competent immune system, because these receptors are telling the T cell that it's okay. This is one of us. And so we have the opportunity to manipulate these receptors or even these receptors. And I think the future appears to be very bright. So the other thing I wanted to point out is the cancer cell is so smart that it is actually talking to the immune system itself. So the tumor cells can make what is called PD-L1, which is a ligand. It will make that protein and release that protein, and it is directly talking to the T cell. And the PD-1 is the tolerance receptor. And so the tumor is saying to the T cell, it is okay to tolerate whatever is around there. Because the T cell has come to check the tumor cell, and tumor cell is telling the T cell, I'm perfectly fine, you don't need to bother me. So I think this is the kind of biology that is going on in our systems when we have cancer. The other mechanism is the tumor cell is not necessarily talking outright to the immune system, but it is talking after it senses danger. So the tumor cell, T cell interact, and then tumor cell will sense something, and it will then produce the PD-L1 to suppress the tolerance receptor. So there are many possibilities. Another example, in addition to PD-L1, is the CTLA-4. And CTLA-4 is produced by the T cell only after stimulation. So the naive T cell does not have this CTLA-4. It's only after it's communicating with the antigen-presenting cell and it senses there is something is wrong, but it needs to make a decision whether to go kill the cell that has the abnormal antigen or not to kill. So it will produce the CTLA-4 and then communicate further, because CTLA-4 is another tolerance receptor, and then T cell will make a decision. And I leave the lower part out because I think I mentioned the participation of tumor cell there. So without going into detail, basically the interaction between antigen-presenting cell and T cell is now considered similar to the synapses in the brain. So there are a whole bunch of protein that come together and signal together. 10 to 15 protein form a big synapse and communicate. And of course we don't understand how this happens, but ultimately a decision is made by the T cell whether to act or not to act. And I think this is an opportunity for us to divert some of the resources in better understanding of this, so that we can train the T cell to kill the cancer cell, because it's all, the secret is all lodged into this complex. So this is TDM1. This is another approach that I think is our future, where you have the antibody and you have the poison, so to speak, on the left side. So the antibody can take the poison to the cancer cell, and normal interaction is the natural process of the cell when the receptor is engaged is to endocytose everything. So it will swallow the poison and then it will die. And as you know, this drug was very successful in breast cancer. So I'm going to skip because of shortness of time. So these are just my final couple of slides. So we know the cancer cells have a very complex genetic mechanism, or have complex mechanisms. And they basically are trying to overcome survival insults. So in order to win from cancer cell, I think the way we are approaching is not the future. Okay. And with the ENCODE project, with RNA being very important in regulation of the transcripts, I think we have to go back to some of our genetic data and incorporate the RNA interaction with transcripts before we say, this gene is doing that, because it's all being modified. And as I showed you, the single gene or single pathway inhibition is not the road to success for us, and immune evasion is very much normal. So the future in controlling cancer cell is less likely in manipulating the cancer infrastructure. If you keep manipulating it, we're less likely to win. I think if we try to kill the entire mechanism, then we don't have to deal with this reprogramming of oncogene and all the smartness of DNA. So I think the bottom two bullets is the toxic conjugates and train the immune system. And with that, I want to thank you for your attention.
Thank you very much for a very stimulating. Please stay a moment. Is there any question from the audience? Please, Steve.
What's the role of the mutations? So you spoke about KRAS, but it's being mutated in most cancer. What's in your picture the role of a mutation in KRAS? Of the frequent mutations? Right. Do they give a loss of function or do they give a win of function?
So KRAS the normal KRAS is highly regulated. It is active only for a fraction of a second, and then it is quiet. But the moment it is mutated, then it is firing constantly. So that's the advantage of having a mutation. And whether the mutation is spontaneous or it is structured is being debated, but I think it is not spontaneous. The genome is engaging different oncogenes, and the gene editing is very common because of the immune system, like the T cell receptor, B cell receptor. These are all genomic editing products, and so genome can do that. There is a very nice paper in Nature Medicine, and I just give you this very quick example. So there is a patient that is wild type KRAS, and the patient receives cetuximab and has a response, and then it stops responding. So the researchers, I think from Europe, they found a mutation in the extracellular domain. So the cetuximab cannot fit into the receptor anymore. But the panitumumab can fit. So then they gave the other antibody and patient responded. So what that suggests to me is that the cancer cell knew that something is going wrong with the receptor, so it caused the mutation. So either you believe or you don't believe, but I think there is a mastermind somewhere in the DNA that is manipulating the machinery of the entire cell.
Actually, I also think that cells can sense the extracellular space, and maybe there are some regulations, some specific mechanisms that actually can come from outside to inside. I believe that there are specific ways the genome can be changed, not only by chance, but maybe by induction from some signals from the outside. So I think it goes along your line that maybe there are some mechanisms to discover still whereby the genome can be specifically modified, not only by spontaneous mutations, but maybe by induction of mechanisms that will indeed provoke changes in the DNA. Could be.
Yeah, I think GIST is another example of with one mutation in one exon, and then you treat, and it gets new mutations.
Are there other questions?
Thank you very much for your talk. I have two comments. First one, you said that the cancer genome evolved over thousands of years. I mean, since then I'm thinking, how would I describe a cancer genome? And I would say, if you take an helicopter view and see all the patients, there is not really a cancer genome, because each patient has its own mutational makeup. So would it be possible, or would you agree to say that the concept of cancer evolution has adapted, and so it's something. Is there something beyond the individual mutations which cannot be detected necessarily by sequencing all the tumors, because each tumor is different? So there is an overexposed general concept where all the cancers end up independent of how their genome look like, and if you would agree, then this would mean that any hypothetical tumor in a healthy person who has not a tumor yet, so the first mutation already would happen in agreement with the immune system, which brings me to the rationale, why then try to train the immune system if it anyway agreed the cancer to come up, because it was never gone, basically. Thank you.
So I think I misspoke about the cancer genome being evolved over billions of years. I meant to say normal genome. But the cancer is using, most of the genome in cancer is normal. There are only a few hundred mutations. or maybe even if there are a few thousand mutations, most of the genomic structure is pretty much normal. So it's using that machinery. So I think I misspoke there. And the other issue is if the immune system is tolerating the beginning of the cancer, if the cancer is here and there is a process and it's tolerating the whole process to the extent that now you have cancer and now you have a big mass. I mean, I don't understand, but I think it is based on the evolution of autoimmunity in us. And so, you know, that the MHC1 complex is also expressed in cancer cells. Some cancer cells lose it, but most of the nucleated cells have that complex. And they are presenting some of the normal native protein. Well, the immune APC, when it comes to check that cell, the MHC molecule is at tolerance. Even APC will not go any further. It will just leave it alone because it's like all other normal cells. So I mean, I don't pretend to understand that, but I think what we understand is what you just said. There is this tolerance to production of a cell that is sick. And immune system doesn't tolerate sick cells. You know, if you infect the cell with viruses, it will come and kill it. If there are bacteria, it will kill it. So, or if it's old or there is something else is wrong, there's a lot of DNA damage, it will just kill. Immune system is cleaning up our body. But the cancer cell somehow is tolerated. Maybe we produce a lot more cancers than we actually experience. But obviously, there is a lot of cancer out there, and the immune system is tolerating. I don't understand.
I think we stop here for the moment, and we will continue at 2 o'clock in pathology. That's the kleiner Hörsaal Pathologie auf dem Stock C. Danke vielmals fürs Kommen. Thank you very much.